I would like to tell you about Ensoma and give you an update on our company. Let's see here. So I don't have to tell everyone in this room about really the great advances that our industry has made with now approved therapies with curative effect in genetic diseases and oncology, and really transformative promise. But at the same time, this ex vivo paradigm really does limit the number of patients that can benefit from these therapies, right? There's really a lot of significant patient burden to receive these therapies, a long patient journey, complex manufacturing, such that it will only treat a fraction of those who could really, benefit from these therapies. So with that, the vision of Ensoma is to enable in vivo cellular engineering that addresses all these limitations of ex vivo therapies by combining our first-in-class genome engineering and delivery. Ultimately, in a one-time dose with a simple outpatient procedure that will allow us to achieve durable in vivo engineering of hematopoietic cells. We have a great team. I'm not going to get into details, but we have great tech ops that are manufacturing this up to clinical scale to generate smart immune cell medicines, both for oncology applications as well as in genetic disease. Our mission is to develop one-time in vivo treatments that precisely engineer any cell of the hematopoietic system to cure diseases from within. We do this by harnessing the power of the hematopoietic stem cell. By engineering that hematopoietic stem cell, it gives us the ability to leverage the native biology of all the daughter cells that we choose to ultimately express from in the blood and immune system. And depending on our application, we've also started to show that we can engineer circulating mature cells as well for immediate effect. And I'll show you some of those data. We do this with our Ingenious system. It's a combination of our engineering, where we have class-leading proprietary A and C base editors, in combination with a high-efficiency transposase system that gives us the capabilities to go from single nucleotide changes all the way up to large insertions. We deliver this with a recombinant virus-like particle that's based on a helper-dependent adenovirus system with a tremendous capacity in nuclear delivery that in combination allows us to generate these in vivo genomic medicines, ultimately in a fully outpatient setting, off-the-shelf single dose for durable cures. So the virus-like particle that we use is based on helper-dependent adenovirus. These are vectors that have evolved over hundreds of millions of years to not only get DNA into the cell, but deliver it specifically to the nucleus. You may be more familiar with first or second generation adenovirus with like one or a couple of genes eliminated. In the helper-dependent adenovirus, all of the adenovirus genes have been eliminated. And this gives us this unprecedented 35 kilobases of capacity. You can see in the bottom right how this compares to other viral vector delivery systems. It also reduces the immune response to our vector transduction. We've also made this using a chimeric VLP with a CD46 binding AD35 fiber that we further engineered for greater affinity and better delivery. We think that the greatest power is a combination of base editing, gene editing, with gene insertion, not either on its own. We think that we're able to achieve more in this way. And I'll talk a little bit more about our base editors that we combine with our Sleeping Beauty-based transposase system for a variety of approaches. So as part of our Series B financing, we acquired a company based in Copenhagen with Cas12a based gene editing tools. And it really is really ideally suited for our in vivo applications. I'll go over three main things, three main characteristics. One is the exquisite specificity. This is actually just for demonstration, a fairly dirty guide, but showing that Cas12a normally has a lower tolerance for base pair mismatches, so a reduced number of off-target effects. And we've engineered these Cas12a-based editors to reduce that further still. We've developed base editing technology that does not nick the DNA. There's actually been growing concern and growing data to suggest that even just nicking the DNA can cause indels, can cause chromosomal rearrangements. Some publications from earlier this year show that as well. But we've been able to achieve, without nicking the DNA, to still achieve high degrees of base editing that we think is really important for in vivo applications. And even more so because we think it's also important to have the ability to multiplex. And here again, a Cas12a-based system using its RNase activity allows us the ability to actually process multiple guides off of a single transcript. And so this is more efficient using a single promoter as opposed to, say, a Cas9 system, which would require separate promoters for the Cas and for the various guide RNAs. We obtain these base editors primarily, obviously, for our own use, but I should say that as we thought more about this, we really think that more companies and potential partners could benefit from this technology as well. And so one such example is within a lot of companies interested in editing based on AAV. Of course, the normal industry standard Cas9 base editors are a little bit too large to fit in a single AAV. They have to be split, which results in some inefficiencies. We feel that what we have with our ingenious base editors, because of that smaller protein and CRISPR RNA and that single promoter transcript that self-processes, that we actually can have a best-in-class base editor that will fit into a single AAV. Now, I want to talk to you a little bit how we apply this first in oncology and how our approach is differentiated. First off, it's in vivo, which removes all of the barriers that come with the current ex vivo treatment into a simple, scalable outpatient environment. It's durable. We can access immediate effect from transducing some percentage of circulating immune cells and follow that with immune cells derived from the hematopoietic stem cell that we engineer, and after a period of time, in the case of, say, NK cells or monocytes, that they come up within a couple of weeks from those HSCs to provide a durable and functional persistence. With our system and that combination of capacity and engineering, we can actually achieve a multicellular approach. For these complex cancers, we think it actually may require different cell types to work in conjunction with each other to really be effective at fighting cancer. And using our system, we can achieve this multicellular approach in a way that most other people probably haven't even thought about, just because it hasn't been possible. Finally, again, because of our capacity, we can make this combinatorial. We've seen a lot of advances, actually, in the ex vivo world where they're combining, a CAR T insertion with certain gene edits. As we look to our system, because of that combination of capacity and the ability to use a transposase to insert some parts for durable expression, as well as have other parts outside the transposon that just have transient expression, let's say with our base editors, we can combine those therapies in a way to leverage those advances in a way that no other in vivo gene therapy can. So let me give a few examples. This is all from our in vivo engineering of multiple cell types. So some examples from direct PBMC engineering, creating CAR T cells or CAR NK cells. And those percentages, if you calculate out the math, actually correspond to what are given current ex vivo cell doses. We can also engineer the HSC, and from those, we can derive multiple cell types. Just give a couple examples of CAR-Ms or CAR-NKs coming out of those HSCs. We've shown that these are functional. The CAR T cells we generate in vivo can expand, and they can also show that in these animals where we're getting this CAR-mediated B cell depletion of a target cell, if we use, let's say, a B cell target. Now, in genetic disease, as I said, we really want to address that patient journey. Right? We want to avoid this myeloablative conditioning that requires long hospital stays and people really rearranging their lives. And it removes all the complexity of the ex vivo manufacturing. Our vision is ultimately to have an in vivo patient journey that's fully outpatient without requiring these long hospitalizations, without toxic pretreatments, with a simple off-the-shelf injection and durable effect. And I would say in our Series B rounds, we found a partner that shared this vision in the Bill and Melinda Gates Foundation, who participated in the round, and we're working together. Obviously their vision is to enable us to get these same curative effects to sub-Saharan Africa, where it is very impractical for some of the ex vivo therapies to achieve that. Now we can do this in a couple of ways. One is with gene insertion. And here we show some of the data where we've inserted a gamma globin gene to correct the defect in a beta thalassemia mouse model, where we can get high levels, almost 100% of the cells expressing fetal hemoglobin. And we can show, as you can see on the right, that this can cure that can correct that anemia in that beta thal model, basically reproducing our original founder's results, which is actually kind of nice because not everyone reproduces their founder's results. So that was good to see. And also we've confirmed through secondary engraftment that we truly are hitting these long-term HSCs. Now, of course, as I mentioned, we now, with our base editing tools, we can approach in another way. And so that we've now applied, and we've now generated some really exciting data for HbF reactivation, where in HUDIP cells we can show we can get high levels of gene editing, and that hitting this particular site that's found with hereditary persistence of fetal hemoglobin, we could achieve high levels of HbF reactivation in a ways analogous to the CRISPR Vertex approach, as opposed to our prior approach, which is more like the Bluebird approach. Also for rare diseases, we can show using those base editing tools, we can cause a protein knockdown, where at the genetic level we can, for example, mutate a promoter that leads to the target protein being eliminated. We can do it through exon skipping, et cetera, all the same things that others have done in the field. So with that, I would conclude by saying, it's really exciting to be building what we think is a premier in vivo cellular engineering company. We have this breakthrough platform. Not only do we have a number of genome editing capabilities in hand, but using our system, in part because of the capacity, we can take advantage of all the advances we see. We see there's a lot of different gene editing tools that are coming up in the future. When they're ready for prime time, we know that they're quite large in many cases, multi-subunit, and this is all going to be compatible with our system. So we're really excited about that in the future as well. We have expansive opportunities in oncology, genetic diseases, ultimately in autoimmunity and other areas. And we feel like we're really well positioned to succeed, both with the team, the tech ops that I mentioned, and I should mention that we, after closing a couple of rounds of financing, are well funded to get through our clinical proof of concept. And with that, I will thank everyone for their attention and not hold them from the free alcohol our prior speaker offered.